Hello, my name is Petya Tatarska, and I'm proud to present to you Innova BM's plant biotech active ingredients. First of all, I would like to tell you who Innova BM is. We develop and produce extraordinary active ingredients through advanced plant biotech. Innova BM is a Bulgarian biotech company which is a world leader in terms of scientific innovation in non-GMO plant biotechnology, headed by Professor Pavlov, who is a member of 2 European Academies of Sciences. Our R&D team boasts some of the most experienced scientists and practitioners in secondary metabolites in plant in vitro culture worldwide. We are a comparatively young company, coming on the market in 2019. Despite the global slowdown due to the COVID pandemic and the Ukraine war, now we are selling on 5 continents, with the US and Japan being our strongest markets. The most important feature of our ingredients is that they combine natural origin with synthetic level efficacy. Also, they produce a truly holistic effect. Each of our active ingredients deliver exceptional potency in their primary effects while offering a wide range of additional cosmetic benefits. Our unique active ingredients truly stand out. They incorporate all the essential features that consumers value. First of all, and most importantly, they work. They produce visible effects quickly. Apart from that, they are from natural origin, they are innovation-based, they are based on patented biotechnology, produced and developed by sustainable, waste-free processes. They are biodegradable, minimalistic, green, vegan, clean, cruelty-free, and Cosmos approved. We are proud to be among the top 1% of companies in terms of sustainability. Our claim to sustainability leadership is supported by 3 pillars. First of all, non-GMO in vitro plant technique in general is a very sustainable way of plant production. It eliminates many of the negative impacts of traditional agriculture, such as soil depletion and the need for pesticides, herbicides, and fertilizers. It saves a lot of energy and water. While the first pillar is shared by all companies working with non-GMO in vitro plant methods, the other 2 are specific for Innova BM. Our products are produced in a completely waste-free manner. Also, we have developed and patented a method of in vitro production of endangered plant species, by which we contribute to biodiversity conservation. I'd like to do a short introduction into plant biotech, which will help you understand our products and how they differ from everything else on the market. Plant biotech is mostly associated with genetically modifying the plants in order to enhance certain desirable qualities. There are, however, also a number of non-GMO techniques. Some of them, like micropropagation, are directed towards helping with cultivation of whole plants. Other methods are applied when the subject of interest is specific plant metabolites rather than the whole plant. This is the so-called plant culture, which can be de-differentiated, consisting of individual cells or cell conglomerates, or differentiated, consisting of specialized plant tissue such as root, shoots, or other body parts. The latter 2 areas are where the focus and expertise of Innova BM are. Let me show you how dedifferentiated culture, more popularly known as plant stem cells, is created. Parts from the plant of interest are removed, and those explants are carefully sterilized and transferred into a nutrient medium, where they start forming dedifferentiated cells or stem cells at the place of the cut. These cells are isolated and stimulated to divide and multiply on their own. This is the so-called Callus, and it is the first step when we can claim we have a stem cell culture. Some stem cell-based active cosmetic ingredients on the market are just Callus extracts. However, transferring the Callus into a liquid media allows the process of optimization to take place, in which the stem cells are stimulated to overproduce the valuable secondary metabolites. As a result, the natural medical and cosmetic properties of the plant are considerably enhanced without genetic modification, which improves the quality of the active ingredient while preserving its naturality. This is the same process illustrated with real-life pictures from the plants through the explants, then Callus, and the most important part, the optimization process. And this is an illustration between dedifferentiated cells and differentiated cells. The stem cells have the ability to turn into another type of cells, while the differentiated cells have a specific function in the plant. They are either part of the root, shoot, and so on, and they cannot transform into other type of cells. So, this introduction was about non-GMO plant biotech in general. Now I'd like to explain what are the advantages of Innova BM's active ingredients. Our stem cell-based ingredients stand out with the remarkable stem cell optimization, high concentration of exosomes, non-extraction biomass processing, unique phytochemical composition, and minimalistic preservative-free formula. The optimization performed by Innova BM scientists increases considerably the concentration of secondary metabolites, the substances that carry the main cosmetic function in the cells. Here are examples from our cultures, before optimization, after optimization, which demonstrate the dramatic differences in secondary metabolites as a result of Innova BM's optimization process. Another advantage of our stem cell ingredients is that they have a high concentration of exosomes. Exosomes act as natural carriers, delivering growth factors and proteins to boost cellular regeneration and enhance cell communication. Recent studies highlight that plant exosomes are improving the efficacy of the active ingredients by facilitating targeted delivery and enhanced penetration of beneficial compounds into the skin cells. All our stem cell ingredients have a high concentration of exosomes. Take, for example, Innova stem cell calendula. Through the method of dynamic light scattering, we have proven that the concentration of exosomes is 3 times 10 to the power of 10 per millimeter, and that the predominant size of the particles is 183.1 nanometers. Another advantage of our plant stem cell actives is the unique phytochemical composition. It is partly due to the process of deoptimization and the increase of secondary metabolites inside the cells, but it is also a result in how we process those stem cells into active ingredients. We have a proprietary non-extraction method of processing the stem cell biomass, which releases the whole content of the stem cells into the active ingredient. Non-extraction processing not only ensures waste-free production, but keeps all the wealth created by nature inside the active. Thus, the synergy created by nature is preserved and enhanced. As a result, we have fully natural and preservative-free actives which are packed with exosomes, secondary metabolites, peptides, exopolysaccharides, intracellular vehicles. I want to explain why our non-extraction process of releasing the whole content of the stem cells is superior to drying and powdering of the biomass. When the cells are dehydrated, this results in changes in the structure of some phytochemicals. These changes cannot be reversed in subsequent hydration, and this affects the efficacy of the active ingredient. And now, the unique advantages of our differentiated culture. Innova BM has developed and patented next-level biotechnology, which is particularly suited for differentiated culture. It allows the mass production of rare plant natural substances of great value. It allows high concentration of beneficial phytochemicals in the cells, and also allows the capturing of exosomes produced by the cells, and ensures the possibility of fully natural and preservative-free actives. Our next-level patented biotechnology was invented with the purpose of developing an in vitro culture from the rare and very valuable plant Habellia rhodopensis. This patent is pending approval in Europe, USA, Canada, Australia, China, Japan, India, South Korea, Israel, Mexico, and Brazil. This next-generation technology was later applied to 4 other rare and protected plant species in the same family, and also covers their antimutagenic, anticlystogenic, UV protection and gamma protection action, and the use as chemo, radio, and UV protectors. This table demonstrates that the in vitro culture of Habellia rhodopensis is richer in beneficial phytochemicals compared to the natural plant, once again highlighting the advantages of our technology.